There are multiple ways that sows can become infected with PRRS virus, including inhalation of aerosolized virus, as well as through virus-contaminated semen. Once the virus enters the sow's bloodstream, it can cross the placenta late in gestation, approximately day 70 of gestation onwards. The maternal-fetal interface is composed of 6 layers of tissue which separate the sow and the fetus. How PRRS virus is able to cross this interface is still not fully understood. One hypothesis is that PRRS virus infects macrophages near the maternal-fetal interface. But how the virus is able to reach the fetus is again still unclear. However, once the fetus has become infected with PRRS virus, it can result in abortions, stillborn or mummified fetuses, or weak-born piglets. How can reproductive failure be prevented, and what parts of the immune system are crucial for the prevention as well as the control of PRRS virus infection? To answer this, let's take a closer look at the virus. The virus attaches to and infects the porcine macrophage. The cell surface protein CD163 has been shown to be necessary for infection. The virus is then internalized. Following uncoating, the free viral RNA is translated into non-structural proteins to support viral replication, and then structural proteins. Virions are then assembled. The virus also infects pulmonary intravascular macrophages, gets into the bloodstream, and spreads both within the lung and to distant lymphoid tissue, where it goes on to infect more macrophages. Several weeks after infection, neutralizing antibodies are secreted from plasma cells. These antibodies bind to proteins on the surface of the virus, ideally preventing infection. However, the ability of PRRS virus to mutate these surface proteins results in the capability of the virus to go undetected by antibodies that were developed against a different strain of the virus. While antibodies recognize extracellular virus, recent research in PRRS virus has shown that antibodies cannot identify infected cells. Via the MHC complex, viral proteins, or antigens, are presented on the surface of specific white blood cells. T cells are lymphocytes that can recognize antigens that are within cells, as they are presented to T cells via MHCs. It is through the recognition of these presented viral proteins and the subsequent secretion of cytokines, such as interferon, that T cells affect the immune response. These cytokines inhibit the replication of PRRS virus in infected macrophages and induce the macrophages to present more viral particles on their surface. Additionally, T cells have the capability to recognize more diverse strains of PRRS virus than the antibody response does, thus allowing for possible heterologous or broad protection and the subsequent limiting of disease. Recent research suggests that a subset of T cells, known as cytotoxic T cells, could be important for clearing PRRS virus infection. Following antigen recognition on the surface of infected cells, cytotoxic T cells release cytotoxic granules that activate apoptosis, or programmed cell death. This method of inducing cell death is targeted only to infected cells and spares uninfected cells. It also prevents rupturing of the cell and further spread of the virus. In contrast to other immune cells, memory T cells are viable for extended periods of time and act as sentinels against reinfection. When they come into contact and recognize viral protein in an infected macrophage, they proliferate and kill the infected cell. Through memory cells, immunity is maintained long after vaccination or infection. To protect the sow and the fetuses that she carries, the sow's immune system produces T cells, which play a central role in the direct, effective response to infection. Through the production of specific cytokines, these cells are able to modulate immunity, limit viral spread, and control the intensity and the duration of the immune response. T cells can eliminate individual infected cells, helping to curb viral propagation and spread to other cells. Finally, while not as specific as antibodies, T cells can recognize proteins which the virus uses to replicate, thus allowing T cells a greater breadth of reactivity to the ever-evolving PRRS virus.